All right, everybody. So thank you so much for listening in on this lecture and being online. Sorry we had that last minute kind of change up between in person and then online, but I'm sure some of you, especially the Friday lab going into break, will appreciate that. So I thought it would be a little bit easier so you could reflect on what we go over. You're not frantically writing notes because it is a lot of more visual, looking at some examples of how to make figures better and nicer, how to use, or at least get a little bit of insight into what R looks like and what coding is, and maybe do a little bit of statistics. So thanks everybody for being here. Thanks for listening, and let's go through it. So this is our last lecture. We'll be going through RStats and a little bit of a course wrap up. So as we look on the lab schedule, we are in week 13. We are in November 19th. No lab next week, obviously, because of Thanksgiving, and then straight into the final practical. And we'll get a little bit into that at the end. Hopefully you saw the announcement. I included some information about the final lab practical in there. So go and look at that if you didn't read the rest of that announcement. It was under the same announcement that talked about today's lab. So I will fill you in a little bit at the end about that. And then after, there's no lab. It's the last day of class. So hopefully by that point, you're studying for the rest of your finals and projects and getting everything done and wrapping up into winter break. So let's get into this. So we're going to be talking a bit about coding in R, especially in R Studio, and we'll talk a little bit about what that means. But first off, what is coding, and what are the benefits? So you might think, oh, this is wildlife techniques. Why am I trying to learn what a computer language is or what coding is? What are some of the benefits? Why are we here? Why are we talking about this? And I would put this as, especially more and more modern, this as a wildlife technique. A lot of times you think of wildlife techniques and it's getting out and capturing, netting, banding, all the things that we've talked about and done throughout this semester. But this is becoming more and more something that people are reliant on, and something that people expect you to know, especially in the wildlife sector. So coding is pretty simple in terms of what it is. It's just telling a computer what to do. But you're getting a little bit more into the weeds. You're actually writing this language. So you're talking to the computer and telling it in some kind of language, what are you going to output for me? And basically that computer language is just what you think about on that right side of the screen. As you're writing different code, it's what the computer or the program that you're using, what it can actually read to then output what you want. And so if we're working in R Studio, which we'll talk about in a little bit, that language might be completely different and might not work with some other program. So it really is a language. You don't just learn one code language and then know them all or know how to code. It's very program dependent, but you'll see that once you learn kind of one language, it might work with a lot of different outputs, and you can do a lot within just one program or one language. So it can help automate repetitive tasks. It removes that human error, especially with different calculations or statistics, but this also can include human error when writing code. Coding is very finicky, and sometimes you'll be writing and accidentally put in something wrong that might mess up what your output is. And so although it removes the human error in calculations, it can induce human error when you're writing code, which is always why you should have people kind of check or double check, or run it through some kind of program to check that your code is working. And a lot of times that just means troubleshooting it and looking at the output and getting it to what you want. It really does let you work quickly with big data sets. This is probably one of the biggest things that coding is helpful for. And you can build off what others have written for your own work. And what I mean by that is, a lot of times if someone has already written an entire code about how to create home ranges with GPS data, then you can go in and look at that code and then build off what they've done for your own work. And so a lot of times you're not just starting from scratch and starting from, okay, I need to build or write this code. A lot of people are out there, and a lot of people have probably done what you're planning to do, and you can use, especially with making sure to cite them, but use what they've kind of learned and gone through and written to then build that into your own code and your own work. So why do I care as a biologist? Well, a couple reasons, as I kind of talked about. It's extremely helpful for large data sets. So think about our bird banding or our camera trap images. If we go through and we have thousands of bird bands that we need to look at, maybe we want to look at mass for a couple different birds, or maybe we want to look at the mean distribution of wing chord or wing measurement for all of the Carolina wrens. We wouldn't want to do that by hand and add it all up. We want to do that pretty quickly or easily, and this can be done very easily in something like in our studio. It also ensures this transparency and reliability for others. So especially others in your field. So then they can look at your code and be able to say, okay, this is what they did to calculate this output, or this is the code they ran to create this figure or plot. And that can be super helpful because one, you're helping them, but two, then it builds reliability or trust or integrity for what you're doing, instead of something that nobody else can repeat. It also allows quick incorporation of new types of analyses. So let's say you run a bunch of, again, home ranges for a certain bird species, and then actually somebody comes out with a groundbreaking paper that says, actually, this is a better analysis to run your home ranges, so then we can better, estimate what that home range is for that animal. Well, maybe you're going to want to run their code with your data to then get those updated estimates. And then finally, it makes processing and visualization much quicker and easier. And so just kind of on the right, again, just a quick plot that would be taking 15, 30 seconds, something like that, if you had the right code, and just running your data, and you can see, oh, we have different groups of penguins, and they have differences in bill depth and bill length. And what are the dimensions of that? And so it can be very easy to just process and visualize the data to then help you and the public, or who you're presenting this to, draw conclusions about your work. So what is R and RStudio exactly? You've heard me say that a couple times. Well, it's a free open source computer language. So R is the language. So you're kind of learning and writing in R. And this is designed for data analysis and also statistics. RStudio is the program that can help organize and run the code you actually write. So when you see me working in a few slides in RStudio, you'll see that I have code that's written, but then I'm running it through this program that you can just download and look at. It's very popular for making plots and figures and also running these statistical tests. It's very widely used in ecology and biology, so this is the one I pretty much 99% of the time see people working in this field, but it can really be used in any field. This is just the one that's very popular in our field. You can process spreadsheets like CSV files, you can process text files, GIS files, so kind of what we talked about the one day with GIS, like shape files or GPS points. And I think the popularity also kind of comes up with R and RStudio because they run through Windows and Mac and Linux. So something like ArcGIS doesn't run on Mac. And so that's why you have to find, if you just use primarily a Mac and you're trying to make maps, you might need to find some other output like QGIS, versus R and RStudio run on pretty much all devices. So what are we actually going to go over today? Well, we're going to look at what RStudio actually looks like. We're going to be loading, cleaning, and exploring some of the camera trap data. We're going to be running simple statistics to answer some questions. And then we're going to be really focusing on making clear plots and visualizations. So just kind of these 4 steps. So first off, we have to talk about data gathering and structure. Now, I know we've talked about this quite a bit, so I'm going to make this pretty brief. So data and data sheets, remember when we're creating a data sheet, or before we even collect any data, what questions do we actually have? What data do we need to collect to answer our questions? And then how might that data look like? And this is the 3rd step that we need to think about when we actually visualize and process that data. So it's usually very project specific, and it can require a lot of information or very little, depending on what your objective of your question is. And the people that didn't come out to bird banding at ENRTA, their assignment was to kind of make a mock research question and objective about a certain bird species in Texas. And it was a very good exercise. And although it's not for points, I'd recommend all of you to go and do that exercise at some point, even outside this class, because it's very good to think about what research questions you might have, what data you're actually going out and collecting, and then how might you actually visualize and process that data. There are many data sheets out there as well that are already created. So work with others in your field of study. You don't always have to make up your own data sheet every single time you go out and collect data. If you have something very specific, you probably need to. But there are a lot of data sheets out there from people that might mirror what you're collecting and might help kind of give you a baseline to build off of. Now, some permits may require information to be collected or reported. So just keep an eye on that. Make sure that you're not leaving out any information that you need to collect. And then also make sure to include those abiotic factors that also may be important. Something like weather, the location, the time of day, things that kind of incorporate all of the detections or the rows that you're going to be writing down. So a very typical data sheet structure a lot of times has kind of the top that has information that corresponds to the entire sheet. So location, date, environment, weather, how many people are out there, the time. Then there's columns for information that corresponds to each of the rows. And so that might be things like band number, species name, that sort of deal, where they have full kind of columns that each of the rows can then fill in, which does bring up the question, what do the rows represent? I'm going to give you a couple seconds just to think about what each row represents. Hopefully you thought about it for a second, but what do each row represent? Well, that represents maybe the individuals, so each bird we're banding, or the detections, each camera photo in a camera trap. These are just kind of our detections or our individuals that we're writing in that we can fill in with the information we're putting in the column headers. But what other information might we want to add to the data sheet? Because there's a lot of other stuff we could add. So here's my Wilson Snipe banding sheet, and what information do we see on this data sheet? Well, it probably mirrors very similar to some other bird banding data sheet, like the one we used out at bird banding. We'll have a spot for band number, a spot for bander initials. We have our kind of things that work with all of the detections or all of the individuals banded on this sheet, like our location, our site, how many observers. But I've added page number to keep everything in order. I've added kind of the banders and the recorders down here. I have a lot of information about different codes that I'm putting up into my actual banding sheet. I also have a place for notes. And so these are all things that you can bring in to help gather and make sure that you have information once you go and visualize and process it. But there are things that I had to specifically put for this data sheet, like the transmitter number for the transmitters I'm putting on the different snipe that I'm capturing. So let's do a little bit of an example. Let's make a data sheet. So for our camera trap example, let's say that you've already placed camera traps at the deer feeding stations, like last week's example, and we're ready to summarize and visualize the data. So we have hundreds and hundreds of photos from our camera traps. Now we need to summarize that data. We need to actually input it. So what are the columns we might need to record from the camera trap photos? So let's create a data sheet and enter a few. And I'm going to give you a couple seconds if you want to pause the video right now and just jot down a few different column headers. And again, column headers are this, like band number, transmitter number. What are some things you might want to collect when you're going through and looking at the different photos of the different camera traps that we set up? So pause the video right now and take a second to jot down a few. Okay, so hopefully you jotted down a few. Hopefully you paused the video. And so I've written down a couple of them. and maybe they mirror what you have, maybe they don't. So I've written down a couple things like maybe we want the camera number. So which camera was it from? Or maybe we just want something like site number. So was it from site 1, site 2, site 3, site 4? That might be something that we want to keep track of. We also probably want to keep track of photo number so we can reference back to it. Maybe there's a note that says, very foggy or something, and we want to keep track of that. We want to be able to put that back to which photo number it is, that unique identifier. We also probably want date, so which day was it, and also time. If we want to track when the deer are coming to the feeder, we want to make sure that we're writing the time down. We also probably want something maybe that is an animal present or not. Maybe we don't want to look at specifically deer. We just want to look at, okay, when is there animals present at the feeder? Regardless of species, just is an animal present or not? Yes, no. And so maybe we have a yes, no. If it's yes, we probably want to put the species. So something like white-tailed deer, or raccoon or possum. And maybe we want the activity. If we want to capture deer at a camera trap that when they're feeding, we don't just want deer that are walking through. So maybe we want to write the activity. Are they staying and foraging, or is this just a quick walkthrough? How many are there individuals present? So is there a bunch of them? Maybe that's the camera or the site we want to capture at versus some other site where there's not a lot of deer. Maybe there's just 1 or 2 that come every morning. And then any notes that we might want to write in as well. So just kind of quickly, if we want to look through some, here we can look at this camera. This is from camera trap 3. And we see, okay, there's 1, 2 deer. This was all the way back. This is old data, so this is not kind of from last week, but this is January 15th in 2025. This is at 3:53 a.m. This is photo 3161. Maybe we want to keep track of the temperature for that environmental condition. So maybe we want to keep track of does the temperature have anything to play? And maybe we want to add that in to what we're collecting. There's only 2 deer present, but they are present. And one's kind of sitting, one's foraging. So maybe we put, feeding or foraging. If we want to go to another one, kind of just scroll through here really quickly. Okay, here it's a little bit warmer. This is January 14th, so I've gone a little bit back. And let's see, we have 1, 2, 3, 4, 5, 6 deer. They're all foraging. That's a lot of deer. So things that we want to keep track of as we kind of fill out our data sheet. And eventually we'll have a full data sheet of this with all the camera traps that we're looking at. Okay, so now we kind of want to go through, let's say we have that data sheet, we have all of the data that we've entered. Now we want to do some cleaning and formatting. And this is really important to do because if you don't clean your data, it's going to mess up your statistics, your visualizations, pretty much everything. So I know it's really exciting to get to the visualizations and get to the coding, but a lot of times you really need to look at your data and understand your data, and you want to clean and format it. So some very common data mistakes. It's very strongly recommended someone proofs the entered data. So if I'm looking at camera traps and I'm filling in the data, I want someone to proof that data as well. And now that can be you. Sometimes you're working on a project and it's just you and you have to proof that data, but ideally it's someone who didn't enter it. It's someone that has fresh eyes, that hasn't looked at the data yet, that then can go through your work and proof it, and vice versa. So some common mistakes that are very common that happen. Maybe you have missing or misspelled data. Maybe the cell is blank when it shouldn't be. There's typos. Maybe there's a duplicate where you just you're looking at a lot of pictures and you just accidentally put in one twice. Or maybe you misspell the date and you accidentally put 1995 instead of, 2028 instead of 2025. Some kind of inconsistent date that doesn't make sense with your data. And how might these cause problems down the line? How can we proof our work besides scrolling through it manually? So just take a second to think about that. How might these cause problems? And I kind of already did say it, is that it's going to mess up our statistics and it's going to mess up our results and kind of eventual visualizations and conclusions. But how can we proof our work besides scrolling through it manually? Well, we can use R. So R is a fantastic source for cleaning and proofing this data. So we can import that kind of CSV or Excel file that we create, and we can check for missing values. We can remove duplicates. We can standardize text and values, and we can also fix data formats or date formats. And so I wrote out just a mock data set that has, about 10, only 10 values that have a bunch of errors. And so you can see that this is, camera 1, 2, 3, and 4, but the date is all weird. This date's input wrong. The times are a little weird. There's misspellings. This says coito instead of coyote. And there's a lot of different missing things. And so this is kind of going through how we can data format and clean it. So just going through a little bit quickly, I'm going to get out of this and we're going to jump into R. Okay, so this is what R looks like. Now, there's a lot of resources out there to talk about learning R. We don't, unfortunately, have the time to go through and teach, how to code, how to work in R. But I just generally want you guys to get away with what it kind of looks like generally, and give you a bit of background, on what it is and what can be done. I feel like a lot of times you might just hear, oh yeah, people work in R, but what can I actually do in R? Is it worth my time in the future to learn how to do this? Is really what I want to get out of today. But I will go through a little bit about R. So I have R Studio down at the bottom. So I've opened R and I've loaded in a bunch of these libraries or packages. And basically, and we'll go through it a little bit later, a package is just a collection of extras that build on the base set of R. So I might not be able to run some statistical analyses if I just open R Studio and start coding, but I might load in a package or a library that somebody's written that has extra, data or coding or language that will help R process that data, or we write the code that then processes through that package that kind of gives us another extra step to work in R. So a lot of times you're loading in a lot of these packages depending on what you want to run and what you want to do. And so I'm reading in my data. This is just in my wildlife techniques R class and I have this cleaning data.csv. I've already loaded in my directory, so it's kind of a little bit harder to explain, but I do have a nice YouTube video if you want to watch this one. Either you can pause right now or watch it later, but this kind of talks about setting your working directory. And basically what a working directory is just like any other program. This is where you're saving, you're exporting, you're taking files in and out. And so over here, I can click files and I can see in all of this one's kind of hidden in my Texas A&M, TAMU grad student folder. And then I've thrown it in my snipe directory because that's my one that I mostly focus on. I didn't want to mess up anything, but I've made a folder that has wildlife techniques R class. And now I've thrown in, some of the CSVs. So if I click this and I view the file, I can view some of the data that I have, but I can also import this. And now we can look at, okay, what is this Excel or CSV file look like when I import it into R? And so this is the data preview, and this is kind of what we saw. We have photo numbers, the date. Oh, some of these dates are wrong. We have some times, some misspellings. But if you had, 10,000 of these, of this data set or these rows, it would be very hard to go through and look at some of the problems. And so we can copy this or just click import and it'll import it in here. And I'm putting this little arrow to then call this entire string, and to call this CSV, I'm just calling it cam data or camera data. So I can write this code up here and I can then click run. And down here on the console, it will tell me that it successfully ran. So I didn't have any errors, and it's saying that it's successfully run through. Now, if I want to look at that data, then I can quickly write in head cam data, and then I can get a little bit, or the first five rows, to look at what that data might look like to make sure that everything kind of came in how I want it to look. Now, for processing or kind of making sure that we clean the data, maybe we want to check for missing values. So we might run a summary. So let's do a summary of cam data to look at, and that's just summarizing what we have. And so most of these won't really help because like camera number or date, there's not really a summary you can have, but maybe we want to look at, number. So our max number of some animal was 6, our minimum number was 1. And our mean was 3. Okay, we have some data. We know that there were animals out there. In terms of kind of looking at these this is looking at unique numbers in species present, or unique values in species present. And there we can see, oh, here's the koi toe. So maybe we have kind of a problem with this one. Somebody's misspelled it. And if you had a big data set, you'd be able to see all the misspellings in there. And so you can use the unique function to look at what's unique in that, certain column. So this is in the species present column right here, and it'll pull out what the unique values are in that set. So we have coyote, koi toe, deer, NA, and bear. All right, now we can standardize these names to help fix it. So here I'm just kind of fixing or coding, and so it'll change when I run this through. It'll run the cam data through, and it'll change koi toe to coyote, and then it'll change bear to bear, coyote to coyote, and deer to deer. So if I run this through, now when we run it through head, oh, we've fixed all the spelling mistakes. So we don't have any more spelling mistakes in the species present, so we won't get any more errors. We can also do that for activity names. So maybe I put moving, and then I put move, and then I put feeding. So I might want to, again, look at these unique data. So I could quickly look at unique cam data, and then maybe we want to look at activity instead. All right. And we have moving, move, feeding, sleeping, feed, resting, feeding. So we have a lot of unique activities here. So again, we want to rename everything to make sure that it all fits. And now let's look at that head cam data again. And okay, now we have moving, feeding, sleeping, feeding. So now when we run this activity in the future for different statistics or visualizations, everything matches up. And finally, we could always fix these data formats, or these date formats, by kind of changing everything into a certain date format so we don't get all mixed up. And I think our date format actually read in quite well. I didn't see any issues with it, which is why it's saying, there's no real issues here. So we should be good on that. And these couple slides are just kind of going through what we just talked about as we kind of clean the data. So this is the old data, putting it in. This is what the Excel looked like versus what it looks like in R. Here's reading it in, removing duplicates, checking for missing data, looking at uniques, standardizing, that sort of deal. So these are all things you can do through R, and there's, again, a lot of different packages or code that you can learn to further clean your data. So now that we have our data cleaned, now we can get into some of the fun parts. So let's start looking at some statistical analyses. So now that we have our data, what are some questions we might want to answer? But we first have to go through what are statistics. Now, I'm hoping some of you have taken a statistics course, or either in undergrad or back in high school. And I'm sure you've probably forgotten a little bit of it, but what generally are statistics? Well, we're just collecting and analyzing data. And this is typically done in large quantities, especially for inferring populations or proportions of a whole from then a representative sample. So obviously our camera trap data we're, getting a lot of camera trap data to understand when animals might be moving, but that's not telling us what all of the animals in the entire research area are doing, but it helps us infer what all the animals are doing from kind of our sample of our camera traps out there. It can help us spot patterns, differences, trends, and observations. Are these differences real? Are they due to random chance? And kind of turning this raw data into usable knowledge for management and conservation. So when we're thinking about our camera trap data, what questions can we ask with that? What statistics can we run, or what are we actually trying to figure out with the data that we kind of went through in our data sheet? So pause the video right now and just kind of quickly write down a couple or think about it for just 30 seconds, a minute. Okay, so hopefully you paused it. If not, do it right now. Make sure to do the exercises. So some of the things that I wrote down were what species are present at each site. So are there deer present? Are there raccoons? Are there possums? What species is most frequently observed? What times of day do we see the most animal activity? If we see a lot of animal activity more in the morning versus the evening, maybe that's when we want to actually capture. Do certain camera traps record more activity than others? Are animals more likely to be feeding or moving? For which times of day? Does the group number differ? Is there only one spot that has, on average, larger groups of animals than other locations? Maybe that area doesn't get as frequent a groups, but maybe the group numbers are larger. So if we're trying to capture, larger groups, maybe we want to focus on that camera or that site for capture versus some of the other ones. So we can do a first look, just looking at some descriptive statistics. So these are summary statistics. These are just looking at kind of summary. So maybe we want to quickly look at, what's the most common species, outliers present. I don't know. What else would you kind of be interested in? But there's a lot of very easy first look we can do on that. And so I'll bring it back over. Bring it back over here. And so to help us run these statistics, I'm going to run basically a bunch of fake data. And so I'm going to set our seed, which is just basically saying, okay, now that I run 42, every single time I run this, it'll be the same data. And we're going to run 120 lines of just fake data. So I'm pretty much just running, let's say it's kind of just mock entering 120 lines of data. So I want camera either camera 1, 2, or 3. I want species present. So let's say deer, raccoon, or possum. But I am putting the probability a little higher for deer because I think kind of mimicking our data, there's probably more deer out there. And I'm also putting the deer numbers larger at camera 1 just so we have something to run statistics through later. And so if I run this, basically just creating fake data for the columns and writing that CSV. So now if we kind of look at that, if we look at camera data, so we look at cam data, now we have new data. And so we'll have 120 lines of the correct dates, the times, animal, yes or no, species present, sleeping, which number, that sort of deal. So just kind of a way to create data for this project. Obviously, it'd be better if we had our own data, but this is just a little bit easier to then show you guys what's happening. So let's say we want to look at summaries. So let's quickly run a summary on that. And that was what we did before. And again, most of our columns aren't numbers, and so we can't do as much with summary, but we can look at date. So we can look at, okay, the minimums, we're now looking at June 6th to June 10th. So this is a date of over 10 days. We are looking at and making sure that all these are 120. So that way we have data for every single column. And then maybe we're looking at number. It looks like the max was 7 and the minimum was 1 in terms of animals present. Let's say we also want to look at frequency. So I'm printing just a quick table, that table function right here, table. And I'm looking at, in camera data, I'm looking at species present, and we see that overall we had 76 deer recordings. So this isn't necessarily the number, this is just species present. So out of 120 pictures, 76 of those were deer, 17 were possum, and 27 were raccoon. We can maybe look at the most common species on the area. So the most common species was deer overall, which we can see that from the table as well. So these are all kind of very simple summary statistics or visualizations just to look at, okay, did we have deer? What's out there? What did we collect? Now we can also compare groups of different, so we can split data into categories. So this might be something like splitting the data into camera location or species, maybe time of day. And this can help compare results to reveal patterns. So like how many deer do we see in camera 1 versus camera 2 versus camera 3? And it's often done with tables or maybe grouped plots. And so if we go back over here, maybe we want to group sizes by camera. So we're going to group everything up. So we're grouping by camera number and by species present. And we can see that camera 1 on average, so this is average. On average, camera 1 had 5.23 deer over the period. Camera 2 had 1.91 deer. Camera 3 had 2.23 deer. So we're grouping the overall sizes and then kind of taking the average of kind of the animals at each site. But this is a little bit harder to understand because it's not visualized. So it's a lot easier to understand when we can actually visualize them. We also might want to look for outliers. So maybe we want to look at, okay, are there different outliers? And we could do that by maybe running some kind of outlier check. And we can see that the outlier group of 7, so one of the places had 7 deer, while the rest kind of were around 1 to 2 to 3. So just looking at and comparing the different groups helped me kind of visualize, okay, we have a lot of deer at camera run. Was there some outlier? Well, there was an outlier of 7, which isn't necessarily wrong, just something to think about. But we can also kind of look at detections by camera. So again, this is basically just not averages, just kind of total numbers. We can see that camera 1 had 30 deer, camera 2 had 33 deer, and camera 3 had 13 deer. So pretty interesting that on average, camera 1 had more deer in a group, but in terms of photos or kind of detections, camera 2 had more. So things that we can kind of think about as we're going through the data. Now we can also compare means. So what is a mean? I'm sure you guys know this. It's the average value in a group. So maybe the average number of deer seen per photo, which was that kind of 5.23 that we talked about. So this is kind of the average number of deer per photo, that sort of deal. And we can compare means to see if there's a real difference. Now there's a few statistical tests to check to see if the differences are likely to be real. And I kind of spoiled it there, but one option we can run is a t-test, which is basically just checking to see if there's statistical differences between the means of two groups. So is it chance that these differences occur, or is it real? And we look at that with a p-value. And I know a lot of you have seen this figure, but a p-value, shaded green area, is the probability of an observed result, assuming that this null hypothesis is true. So if we say, these deer, every single site is likely to have the same amount of deer, then maybe when we see a result that's significant, we know, oh, actually one site ends up having a lot more deer. That's not just random chance. That is statistically significant, that one area might have more deer than the next, or one site or camera is showing more deer than another. So we can actually run this. So we can find all the group sizes for deer at camera 1. And so I'm just taking camera number and then species present, and then I'm taking the number of deer. So I want to get the group sizes. And then I'm looking at the group sizes of all the other deer. And here's camera one. Here's all the group sizes for camera one. So one picture had 3 deer, one picture had 3 deer, 6 deer, 4 deer, 6 deer. And then we can look at the other ones. 2, 3, 2, 3, 1, 3, 2, 2, pretty low. And then we're going to run a t-test to see if this average size of deer is different between them. And that's pretty easy. You just put t.test and you're looking at camera 1 versus the camera 2 and 3. And we can see that when we pull up this t-test, our p-value is very significant. It's very, 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 very low. But if you want this in plain language, you can write just a little code that says either there is or is not at a p-value of 0.05. And if we output this, there is statistically significant difference in the deer group sizes. So camera 1 is statistically significant and different between camera 2 and 3. Basically camera 1, and we can infer from all of these numbers that camera 1 probably has a lot more groups or larger groups of deer coming to the camera or the site than some of the other areas. So this might want us kind of thinking or help us think, okay, we probably want to catch at site 1 or camera 1 because there's way bigger groups of deer coming. And that is statistically significant versus some of the other cameras that we have out there. But nobody wants to look at this. People want to look at visuals or results because they can help us see patterns quickly. It's easier than reading numbers off code or off a table. And really the important thing is that it makes it simple to communicate findings to others. So it can show trends, it can group differences and outliers. And so we can really show the public or the people that we're trying to communicate this to what the outcome is of our objective. So how can we make effective figures in wildlife science? Well, what makes a good looking figure? These are some things that I've written down and you can look up more online. I would definitely recommend you go through scientific articles and look at the figures they have and then look if that figure looks good to you or not. Some things that you should probably have are clear and informative titles. You should have your axes labeled with units, maybe the legends if they're showing multiple groups. You don't want to have any clutter, so a lot of times I avoid grid lines or unnecessary color. You can have large readable font is a big one. The fonts, a lot of times you need to make sure that they're large, especially if you're putting it on a poster or some kind of presentation. There should be a consistent logical color scheme, and it really should be understandable on its own. So what figures should we use for our different sets of data? Because there's a lot of figures out there. Again, I wrote down a few of them, but you're welcome to look up more. Some things, and these are probably ones you've ran into, things like a bar chart to count frequencies. So maybe detections per species, a line chart to maybe look at trends over time, a box plot to compare distributions. Maybe we want to look at a scatter plot to look at the relationships between two variables, or something like a pie chart to look at the percentage out of the whole, like number of deer counted, broken down by each camera site. And then we want to visualize that spatial data, maybe by a map or, again, by using one of these figures and plots. But in R, how do we actually make these visualizations and plots? Well, there's a handy dandy package and library called ggplot2. And you'll hear a lot of people talk about this. It's a very, very powerful and flexible package for making clear figures. And as I talked about before, a package is just a collection of code and data and documentation that extends the base of functionality to do specific things. So if I want to make better figures than what's currently available in base R, I can load in ggplot2 and it gives you a lot more flexibility to then create these figures or plots that I want. Works with all common data types and visualizations, and it's very easy to customize the titles, labels, colors, sizes, etc. So I threw in a couple of just plots and figures that I've made in the past for a presentation, and so you can see that I have a legend. I keep everything very consistent. So females are blue, males are red. I have a legend. I have migratory speeds and distances. I have speed and distance on the bottom with my units. But again, I'm keeping everything very consistent. the font's large so people can read it? Or what about this one with home range size by season and sex of Wilson Snipe? You can see that the font's large. We're talking about home range area in kilometers squared. And again, I'm keeping those colors consistent with female versus male. So we'll quickly go over back to R and actually go through some of these visualizations. So first I kind of wrote out, let's make a bar graph. So we're going to be using the camera data again, and we're going to look at species present. So what species were present at our cameras? So let's look at it, and I'm calling it P just to keep something easy. But here we go. We've created a plot. We're creating camera data, and we're creating a bar graph. And we're looking at deer, opossum, and raccoon. And we have species dot present and count. Does this look like a good figure? Well, we can clearly see that there's a lot of deer that are coming to the cameras, which is great. But this really doesn't tell us anything else about it. It looks pretty bad, pretty ugly. It's just not a good looking graph. And so maybe we want to make sure that we fix these labels, because right now it's just pulling straight from the columns. So we have that column species.present. We want it to make a little bit better. So let's add some labels. And now we don't see species.present. We just see species and number of detections. We have detected species frequency at the top. This is looking a little bit better, but it's still not that great. The bar graph guidelines in the back are really throwing me off. It's just not a very good looking plot. So we want to add something like a theme. So we could add theme minimal, and that makes it very, very minimal. It makes everything nice and clean for me. And I'm also getting rid of these grid lines. I don't like the grid lines. So let's add this again to our code. Okay. Now it's looking a little bit better. The differences of grid lines are nice, but the fonts are still pretty small. Deer, opossum, and raccoon are very small. Everything's very hard to see. So let's add some sizes to these fonts. So we can look at the different axis titles, like the x-axis or the y-axis, and we're going to make these bold, and we're making them a lot bigger. So if we run this through and add it, there we go. Now we can actually see the fonts. We can see what everything's talking about. It's still not the best graph, but at least it's giving us some information about, detected speakers frequency. And maybe we want to put that, at ENRTA or at cameras 1, 2, and 3. But this is just number of detections for species. How many times are the species being detected at the site? And so now we can start to see a little bit of information. But as a final, how about we add something like manual relabeling? So manual relabeling. So right now we have deer, opossum, and raccoon, but they're not capitalized. So let's recall them deer, deer, raccoon, raccoon, possum, opossum. So let's recall them that. And let's run everything one last time. And now we have deer, possum, and a raccoon looking a lot better. And if we just put stat count, we can actually put the counts on the top. So now we have, again, not the best figure, but something that tells us, all right, how many deer occurrences or frequency did we see at the cameras, and everything's capitalized and labeled and looks pretty good. So this would be something that you could continue to work on and add things to our graph to make it look better in R, but you can see there's a lot of flexibility that you can use. Now let's look at maybe summing number per camera. So right now we're just looking at number of detections, but we're not looking at groups. So it might be 76 deer detections, but not necessarily 76 deer. Because remember, there's a difference between if they're present and then how many are actually out there. So here I'm grouping the species present and summing it with number to create basically a total deer that were actually at each camera. So I'm running basically all of the information we had up here. I'm rerunning it. So I am getting rid of those grid lines, getting the theme as minimal. I'm labeling our axes, and let's run this through. And oh, I missed one. We're up here. Yeah, okay, we're up here. And now we can see at the different camera locations, so we're breaking it up by camera, we're summing the total number of animals. Now we can see, okay, deer, were 157 deer were counted at camera 1 over the study versus only 63 at camera 2 and 29 at camera 3. Now there are still things I'd want to do with this. I'd want to capitalize these again. I'd want to make everything look a little bit better. But overall, now we can start to visualize, okay, now we're seeing a lot of deer on camera 1. This is starting to make me think, okay, we probably want to capture at camera 1. We have a lot of deer out there. Now we can also look at group size by box plot for just deer, because we want to look at, okay, what is the average group size? So maybe we had 157 deer over here, but maybe we hadn't run the t-test yet. We hadn't run anything, and we wanted to visualize, well, how big the groups are for if we want to capture at least 5 deer when we go out there, what's the average group size? So we might run a box plot to look at that. And so if we run a box plot, we can look at, oh, camera 1, the average number of deer at each detection of deer is up to 5, which is great as an average, because now we know if we go out to camera 1 and we go out to that site to capture, there's a good chance if we drop that net when there's deer there, we're going to capture a pretty big group. Versus if we look back here, we had 63 detections of deer at camera 2, but the average number of deer was only 2. And so if we do go out there and capture deer there, we're probably only going to capture 2 deer. So it's probably better to capture at camera 1. Now we can also look at, let's see what else we want to look at. So what if we look at over time for all species? So now we've looked at, okay, we want to capture at camera 1, but what time do we want to capture? So maybe we want to look at animals per day for the species, and we can again group by date for species present, looking at the number. So let's run this and let's plot it. And we can see over the entire study, so this is over all the days, June 10th, the last day, counted the most amount of deer over all of our cameras for total number of animals. But we did have the spike here in June kind of 5th and 6th. So maybe we want to look at those weather conditions and look at, okay, what pushed it to see a lot of deer around June 6th? Was there a big cold snap? Was there a big cold snap here? Was it raining? Do we see those trends in other animals? And in this case, we don't really see it. Something about the deer, but wasn't really keyed in with the possum and the raccoons. Maybe we want to look over the 24-hour period. So we'll again group this, looking at just hour, and then we're going to plot it for just deer and a possum and a raccoon, and look at animals detected by hour. So now we're looking at hours of the day, and pretty clearly there's no kind of correlation, and this is because, again, this data was randomly generated. But if you had real data, you might see peaks between kind of around sunrise and then around sunset when the deer come, crepuscular, come out to forage right before sunrise or during sunrise and during sunset. You might see peaks where the deer are using that feeder. And that might be saying, okay, we should probably capture, or maybe we see a lot of activity during the morning and then no activity at sunset. Okay, we probably want to capture deer at sunrise versus sunset. So these are all just kind of different plots or figures you can make. Again, I don't expect you guys to learn how to code or know all of these plots or figures, but just trying to get a background of these are things you can run through R. And so when you go into the future, just knowing what's available to you. And it really isn't too hard to learn, especially with so many resources online to look at, to watch, to study, to practice. And again, this program is open. This is open source. This is free to download, so anybody can download and just start learning how to do it and working with it. So if you feel really motivated and want to try this out, I'm always happy to kind of talk more about it and kind of do more of a one-on-one program or focused on it, and then also just watching videos and learning how to do it yourself. There's so many data sets out there that are free to use, that are publicly available, that help you kind of work through big data sets through R and visualize data. So hopefully this was a little bit interesting to you guys. I know it was a lot, but I tried to keep it out of the weeds as much and more into this is what you can do and learn and visualize and kind of complete in R. So that is pretty much it in terms of what we'll be doing in R. So if you have any questions, please let us know. Thank you so much for the semester. It was a ton of fun. I loved being able to teach it with Lulu. We hope you guys have learned something today. Please fill out the class reviews at the end of the semester. Again, Lulu and I really tried to revamp this year to try to teach you guys things that were a little bit more modern or things that were maybe a little bit more applicable to what you're going to be doing in the field. And so please fill out the class reviews. It really helps us kind of continue to hone in future years to make sure that we're making this as best a class as possible for you guys. So please talk about improvements or opportunities for growth as future TAs, even if they're not Lulu and I, continue to work in this class. Also, if you have any questions about the practical, please let us know. We're happy to set up private or public days where you guys can kind of come in and look at the specimens again, take pictures. If there's any questions that you have specifically, please ask me. I'm always welcome to talk to you guys and answer. Remember, you can email me at any time to try to set up office hours. I'm always in the building pretty much all the time. Happy to walk you through specimens, teach you, talk about things that we've talked about in the past, review all the slides we've gone through, they're posted, review what we talked about, review the quizzes, review your midterm, because I guarantee you we're going to be pulling things from that. So if you don't have your midterm, please come email me or Lulu so we can give those back to you, either taking pictures and sending them through you email or just coming and grabbing the actual physical copy. But again, please email us with any questions you may have. And deer capture will still happen. It's still currently going to happen. We wanted to give a little more time, but as we said in the announcements, Friday's group where we set up, we had so many deer. I think they had 14 deer on one of the cameras come through at that site. So looks like it's going to be a pretty good capture. We're really hoping for the best, but it's probably going to happen after Thanksgiving. So look forward to that. Should be a great way to kind of end and round the semester out as a kind of big class deer capture, hands-on. So really looking forward to that. And hopefully we get that scheduled soon. So that is it for everything that I have. But again, please feel free to reach out to Lulu and I, and we are here to help you guys and to teach you what we know. So thanks again, and I look forward to seeing you guys when we come back from break.